It's a great day at News Munchies today. Everybody wants to talk about marijuana and its growth in America. Hello, it's Ted Corliss, editor of News Munchies. We continue to focus on identifying important information associated with the growth of cannabis and the reformation of America's cannabis laws. You know, many times we're reached out to by people in the community, the national community, I should say, to address different events that are coming up in the cannabis world. This particular podcast, we have the benefit of talking to some of the individuals that are responsible for the CannMed Fest that's going to be at UCLA October 22nd, 2018. We're joined today on the phone with Doug Kennedy, the co-founder of CannMed Events, and who is one of the directors at Medicinal Genomics Corporation. We'll let him explain that a little more in detail. We also have Benjamin Amaral, who is the marketing manager of Medicinal Genomics. Gentlemen, very nice to talk to you.
Nice to meet you.
Hello. I'll tell you what, you guys are making a big move this year. I understand that the event in October was originally at Harvard, where it had been for the last 2 years. Is that right?
It was, yes. We started at Harvard 2 years ago. Last year we sold out the space that was available there, so we decided to move it over to the West Coast and host it at UCLA in the brand new Luskin Conference Center that holds twice as many people as we had last year.
Fantastic. Well, what I'm going to suggest to our listeners is I want you to stick around a little bit, and I'm going to give you a promotional code that you can use if you intend to attend the event. You know, let me say as a general concept, this is one of the more scientific-based events that I've seen in the community for a while. Would that be an accurate statement?
I think that's totally accurate.
Well, Doug, would you say, you literally, if you go on the website for CannMed, it shows you a legion of scientists and technicians on the issue of cannabis. It's pretty extraordinary. How did you accumulate such a list of people with these backgrounds?
Honestly, there are deep relationships in the science field in the cannabis space. The people that are speaking at the conference, many of them submitted abstracts. We received over 150 abstracts that our advisory board went through. Many of them were incredible, and we then produced a list of what the presentations will cover that you see on the website now. Some presenters on our website are invited based on what we knew they were working on and the results they had achieved, and we knew that would bring great awareness to have that presented at the conference. So we're grateful for all the presenters and all the work that they do in the field, and it's a pleasure to work with all of them.
Well, you've definitely reached out to some of the individuals that are the go-to people in the scientific community on cannabis. And I don't apologize for the fact when it comes to cannabis, I'm a huge nerd. I love getting into the scientific issues associated with it because even as a lawyer, I acknowledge that the science is going to drive a lot of the laws and regulations associated with the expansion of cannabis use. What is it like to deal with so many scientists when you're talking about something that is illegal under federal law, Doug?
We choose our topics based on their recent discoveries. So our scientists, while they are leading the industry in their field, they're extremely humble in how they go about doing what they do. So our approach has always been to bring to the table what we've learned so the audience, people in the audience, in whatever respect they are in the field, can learn from it. But it's a pleasure to work with these people who are set out with a goal of advancing the science and the medicine behind it to better the lives of patients who ultimately will need it to improve their health. So that's the ultimate goal of any talk or any aspect of CannMed.
Now, I understand that by moving your event from Boston to California, you're obviously moving into a far more friendly cannabis environment. Would that be a fair statement?
Yeah, I would say so.
Yeah, definitely. Well, I think that it's the idea that it's been, California, in 1996 is really when we got medical marijuana on the board, and it's been developing over time. And other states like Massachusetts, or obviously educational areas like Harvard, are interested in and continue to grow in the cannabis area. but it looks to me like California is a little bit ahead of everybody else.
Yeah, absolutely, and that's what we saw. And we were based in Boston for the last 2 years because Medicinal Genomics, our parent company who puts on the event, we are Boston based, so it made a lot of sense. And there are a lot of great researchers in the New England area and in the medical community who are looking into this. So we certainly attracted people from there. But what we were seeing, which was a real testament to the presenters that we had and the information that we were providing, we were getting a lot of attendees from the West Coast even to come to Boston to hear the talks. So when we made the decision to expand CannMed and to offer it to more people, we thought it made sense to move it out west, where the industry is really booming and blossoming, and we could really kind of tap into that knowledge base. So it was a really great fit.
Well, it seems like it. It definitely does. Now, tell me about the issue of you're gathering all this information from all these individuals, and these individuals will be presenting at various intervals at the event. Is that the way it's going to work? And then are you going to do anything afterward in terms of this is such a massive amount of information? Are you collecting it and distributing it in any way?
Yeah, absolutely. So we'll be recording each of the talks, and the videos will be available after the conference, but we still encourage everyone to come out because, beyond just the information that's being shared, there's the opportunity to meet with what we're calling, and we truly believe are the greatest minds in cannabis science and cannabis research. They're all going to be at UCLA this fall, and we're really excited about it. And one of the things that we're looking to offer in 2019 to any attendees of CannMed 2018 is they'll be automatically enrolled in our webinar series that we're going to be doing. Be a bi-monthly webinar series. So every other month we'll have a new topic presented by some of our alumni speakers and led by our scientific advisor, Dr. Raphael Meshulam, who is the grandfather of cannabis research and a partner and a friend of the conference. So we're really excited to offer that to everyone who comes out this year.
Very nice. Now, Ben, you are a marketing manager for Medicinal Genomics. Can you tell me generally about Medicinal Genomics in terms of when it was formed and what it's been working on these past couple of years?
Sure. Medicinal Genomics was actually formed in 2011 by our Chief Science Officer, Kevin McKernan, who, Medicinal Genomics was really born when he sequenced the first cannabis genome. Kevin is an alumni, or I should say he worked on the Cannabis Research Project, or the Human Genome Project, I should say. So he has a vast knowledge of genomics and, extracting DNA and DNA sequencing, and saw that there was a real lack of knowledge into the genomics of cannabis, and it being more researched and studied and becoming more normalized as a medicine. There was a real knowledge gap there. So he actually went over to Amsterdam with a lab in his suitcase and actually extracted DNA over there and brought it back and assembled it and was the first to publish it. And so that was really kind of what kicked off Medicinal Genomics. Since then, we've developed a number of different DNA-based testing products for the cannabis space, both in the safety testing market and also tools for breeders and growers to breed better crops, increase their yield, and really drive productivity. So that's what we're really focused on in Medicinal Genomics.
Very good. Well, I know that I have trouble coming through TSA. I'm very careful, but I'm assuming when he came back with the DNA, they had some questions about what he had in his briefcase. But
No, luckily there's no cannabinoids in DNA, so he was in the clear.
Got it. Okay, he didn't have to declare that. I understand. When I hear things in other environments associated with genetic research and the development of different ways to treat disease, it certainly seems to me it makes sense to take such a dynamic molecule like any of the cannabinoids and to break it apart to understand how it works and how it can be altered in the future. Would you say that you guys that's where you're focused on, is trying to develop bigger and better ways to make marijuana?
In a sense, yes. So what we're really looking for as we're sequencing different strains of cannabis is trying to identify genetic markers that could be markers for, like you said, different cannabinoid synthase, different cannabinoid profiles, but also more agricultural type markers, flowering time, resistance to pesticides or pests, I should say, being more hardy for outdoor growing. These are the types of things that are really going to drive the market and kind of set cultivators apart. The ones who can sort of identify these traits, breed for them, and innovate are going to be the ones who really make out in the end. So what we're doing is trying to partner with big growers who want to do sequencing projects with us and help to get that information and hopefully share it with the community at large to really kind of push the industry forward.
Understand. Well, I certainly appreciate that and hope that you continue to succeed in that process, simply because I think these kinds of objective criteria, understanding these issues on a scientific level reduces the abstraction, so that when we go to the legislators at various states or even on the federal government to make changes in the country's marijuana laws, that they can learn from the science that you've developed.
Yeah, and that's really what we're trying to do with CannMed, is to really show the research that's going into this, and all the great minds who are working on this, to sort of try to reduce the stigma that's out there that's associated with cannabis. I think a lot of people think that medical cannabis is just a term for people who want to get high, and sure. That's the furthest from the truth, from what we can see from the presenters at CannMed. There's a lot of good research going out there and a lot of great clinicians that are using cannabis to treat a variety of different conditions. We see new conditions every year, and that's what's really exciting, that as we learn more about this plant and more about the different compounds that it produces, we're finding all sorts of different uses for it, and that's really encouraging. And we're really excited for where this industry is going.
Agree very, very much. Would you say that the focus has obviously started out looking at the individual cannabinoid that is cannabidiol, or what we call CBD, but you all are seeing development not just in CBD but in other cannabinoids as well?
Yeah, absolutely. It's interesting, if you go on our website at CannMedEvents.com, one of our recent blog posts, it was an interview with one of our presenters, David Miri, who runs a large cannabis research lab out in Israel, and he was talking about they're doing research into what cannabinoids will actually kill cancer cells. And he tells a great story about they've identified a specific type of leukemia cell that can be killed with a combination of 3 different cannabinoids. And he mentions that they're not the major cannabinoids everyone knows about. It's not THC or CBD or even CBG. One of them actually hasn't even been named in the literature. So that's how little we really know about this plant and how much potential there really is. That there's compounds that haven't even been named yet that are being shown to actually kill cancer cells. So it's really exciting.
Very exciting. And I know also from your website that you have an enormous focus on the neurological disorders that can be treated with some of the cannabinoids from the plant.
Yeah, that's right. And that really ties back into sort of the parent company that Medicinal Genomics was born from. We were originally part of a clinical diagnostic company called Cortigen Life Sciences that did genetic testing for pediatric neurology issues. So testing kids with developmental delay or autism or epilepsy specifically to find out what genetic mutations that they had that were causing their ailment. And as I'm sure you know, CBD is very effective in treating epilepsy. We sort of were seeing that correlation between cannabis and epilepsy, a condition that we were testing for so much, that, we knew there was a connection there. So kind of in the early stages of CannMed, we really focused on that connection of CBD and epilepsy to really sort of further explore why that is and what new products could be developed to help kids with this issue.
Fantastic, fantastic. Well, we're about to take a short break, but before we do that, Ben, I'd like you to go ahead and give us the identifier for your website where people can go to find out more about the CannMed event, October 22nd, and then I'll give them a promotional code. Go ahead.
Sure, absolutely. Everything you need to know about CannMed 2018 is at CannMedEvents.com. We encourage you to buy your tickets and sign up for updates, because we're continuing to add new wrinkles into the event. We have a couple things that we have, a couple irons in the fire, some exciting announcements that are going to be coming out. So be sure to sign up for those updates to stay informed, and we'd love to see you out there this October.
Thank you for that, Ben. And I have an opportunity to, go to different events that are more social, some scientific, some legal. If you have any interest in the scientific issues associated with cannabis, this is an event you really need to take a look at. And if you go to the website, it'll give you a promotional code opportunity, and we're going to give you MUNCH. That's MUNCH 18, MUNCH 18, and you'll get a discount on the tuition to attend the event. This is Ted Corliss with News Munchies. We'll be right back. Hello, it's Ted Corliss with News Munchies. Follow us on our website at www.newsmunchies.com. Also look for us on Facebook, Instagram, and Twitter. News Munchies, dedicated to the legalization, medicalization, and reformation of America's marijuana laws. It's Ted Corliss back here with News Munchies podcast. We have the opportunity today to spend some time with some of the professionals associated with the CannMed event that's scheduled in California for October 22nd. I would strongly recommend you go to the CannMed website, which is CannMedEvents.com, or go to medicinalgenomics.com for more information about medicinal genomics and this particular event. This is, I believe, the third year that they've had the event. The first 2 years it was at Harvard, but they've moved the event to California with the hope that they can draw a lot of the energy associated with the cannabis market in Los Angeles. We have the benefit now of speaking with Kevin McKernan, and Kevin is the Chief Science Officer at Medicinal Genomics. Kevin, very excited to talk to you. I'm one of the people that's very passionate about the genetics, as boring as that may seem to other folks. And so tell me, you're very involved, you have been involved with mapping the genome of the marijuana plant. Is that correct?
This is true, yes. We started on this back in 2011, trying to sequence a strain called ChemDog with the technology we had available to us at the time, which didn't do a very good job, honestly. It turned out to be a very, very difficult genome to sequence. But fast forward to today, we now have tools that can completely iron out this picture from one chromosome tip to the other and give us beautiful cannabis reference genomes, which is one that we just did back in August. We put one public back in August, known as Jamaican Lion. So it's it's the most complete cannabis reference genome publicly available today, and you can download it at our website.
Is that one of Snoop Dogg's strains?
The name makes you think it is, right? But I don't know if the history gets back to him. I understand it dates back to 2007. It won a San Francisco Cannabis Cup back in, I think, 2011, and it's known to be a type 2 plant, which means it makes both CBD and THC, and it's also a beta-caryophyllene dominant strain. I mean, usually type 2 plants people assume have myrcene in them, but this one happens to have beta-caryophyllene. And beta-caryophyllene is an important CB2 agonist. It hits the cannabinoid 2 receptor, so it's a very interesting plant for a variety of reasons.
Well, was there a particular reason why you selected ChemDog? Because I've smoked ChemDog before. Fantastic strain.
So one reason we did it there's a couple reasons. One, the legend that goes back to the Grateful Dead show. That was very appealing to us. But two, we saw it named 3 or 4 different ways on the internet, which made us think, okay, let's just stick a nail in the nomenclature coffin right here, because we've seen it spelled as ChemDog 91, we've seen it spelled as ChemDog DAWG after the Grateful Dead reference to some of the other musicians used to play with. You can see it mutated in nomenclature a variety of ways on the internet. So we figured, why don't we sequence one and just put a line in the sand and begin to assist in that nomenclature problem.
Well, let me back up and maybe talk about things on a really simple level. It's my understanding that one of the issues that you face with trying to normalize or predict how a particular cannabis plant will grow is that the individual plants tend to be very idiosyncratic, meaning that they may not be that much like mom and dad. Is that right? If you take a male plant, cross it with a female plant, you're not necessarily going to get a plant that is reflective of all of the characteristics of the parents. Would that be accurate?
That's right. So that's one thing that actually befuddled the first ChemDog genome we sequenced, is that the mother and father genomes can be almost as divergent as humans and chimpanzees can be and still interbreed. So they can create incredibly diverse offspring, which means the nomenclature system we've had in the field hasn't really captured that. They will tend to name anything from a mother plant, whether it be a clone or a seed from that plant, the same thing. Well, the seeds we know are in fact very different. And unless they have stabilized that line, in other words, they have back-crossed it multiple times, sometimes requiring 5 to 10 back-crosses for you to really stabilize it, what that does is it makes the parents really, really inbred. So when they have children, they're all much more likely to be clonal. That doesn't really happen in the cannabis field. People tend to make crosses, and they're extraordinarily diverse parents, and so you get very diverse offspring. For this reason, you see a lot of indoor grows using cloning to try and stabilize things. It's probably a lot more expensive than making seeds, but it gives them a monoculture of crops that tend to make the same cannabinoids.
Right. You're saying that rather than building a whole new plant, you just take a cutting and then you grow. Or would it be like you were coming off of a mother plant, and then you would just keep cutting her?
Some mother plants might get you 500 or 1,000 cuttings or more, but there is a risk that you run by having something that's genetically identical in your grow. Is that once you have a monoculture, if it happens to be susceptible to powdery mildew or a pest, that's going to spread throughout your entire grow very, very quickly. So there's a balance that we need between having stabilized genetics such that there is some diversity in your grow, but perhaps not something that's perfectly clonal. And you'll see growers playing around with this. It's rare that they'll grow one strain. They'll probably have 8 to maybe 24 mother plants that are their favorites that they're trying to consistently harvest.
If I understand the word clonal, are you saying a plant that would truly be at least similar to the plant that it was cut from?
Exactly, yes.
Okay. But I guess the question becomes, how do you got the issue of, even though the plant may be clonal, then you have to deal with environmental issues that could ultimately affect the growing of the child plant, if that makes any sense.
Yeah, so if you look at very large-scale agriculture, which hasn't really hit cannabis yet, no one uses clones. They all find ways to breed stabilized seeds, which is a process of back-crossing the plant so it's so inbred that when it makes seeds, those siblings are almost like twins. Not quite, but close. And then you can plant millions of those seeds and not have to do a cloning operation. And that's what you see most of traditional agriculture actually use, because the process of cloning is just very manual labor and very intensive at the moment. Whereas making seeds, you can make thousands of seeds from a pollination event as long as you're very careful about the parents you choose in that cross.
A lot of times when I walk into a dispensary and I see a word, let's try one, Girl Scout cookies, which happens to be one of my favorite strains. In fact, I call it my spirit strain, because if I could figure out a way to feel like I've consumed Girl Scout cookies every day, I'd do it. Put a little pack on my hip, and I'd be Girl Scout cookie boy. But there is this idea that I go into a dispensary in Las Vegas and I buy a quarter ounce of a particular plant. Then I go to LA and I walk into Buds and Roses and I buy another Girl Scout cookie. How do I know these plants are going to be even remotely similar?
Well, from the work that we've done is you probably don't. Unless they're coming from the same manufacturer, it's unlikely that they're exactly the same cutting. And this has been showcased in a lot of studies where they do DNA sequencing on all of these things that have the same name, and they find out they're wildly different. And they'll also do chemotyping, which is they look for the cannabinoids and terpenes that are in the plant. And that's probably the cheapest and quickest way to zero in on whether it's the same thing is make sure there's a cannabinoid and terpene profile present on the product. That represents a very small portion of the chemical diversity that's in the plant, but it's probably the most the chemicals we know the most about today. The genetics are meant to fill in some of those gaps. The plants are known to make hundreds of compounds that aren't actually being measured in a lot of the testing labs yet. And so the genetics act as a proxy. Is this at least the identical fingerprint, the identical genetics of the plant? And now from there, you can now that gets you in the ballpark, but you can certainly have clones that are identical and grow them in very different environments and get them to express slightly different terpenes. And so it's rare that they'll switch the cannabinoids that much, but they can alter their terpene expression based on the environment a bit more. And so the genetics kind of get you within a certain ballpark, but really at the end you want to measure the terpenes and the cannabinoids.
Is it your sense that we are developing plants that will be more predictable, or are you all kind of moving away from the plant and focusing only on the cannabinoids?
No, I think we want plants that are reproducible only because we continue to see studies that show full spectrum cannabinoid oils being more productive than isolated compounds. And what that tells us is that we're making a lot of assumptions that THC and CBD are where it's all at. There are lots of other cannabinoids that are being shown to have very potent effects at low dosages. And so we don't yet know whether it's the flavonoids, the cannabinoids, or the anthocyanins that are in the plant. There's a tremendous diversity in there, and I think we're making some assumptions that it's all THC or CBD. So since we don't know exactly which compounds are playing which role, but we do know that full spectrum oils seem to be have more efficacy, we think the best thing is to basically measure as much of that chemical complexity as you can with analytical instrumentation, but to the extent that you can't, get the fingerprints of the plants correct. So you at least have, that you're dealing with the same genetic repertoire. And if you grow that genetics at the same given environmental conditions, you should get the same outcome. That's probably the best that we can do today.
Sure. Well, that's a lot. That's a lot. As I was looking at the website for medicinal genomics, there was one particular product that you offer called Strain Seek. Can you tell us a little bit about that? I'm very curious about that.
So that's a product that has two different tiers. It's a sequencing product. One sequences the entire genome. And that product actually has had a great upgrade recently with this new Jamaican lion reference we have. All that data maps much more beautifully. And then the other one, for those who don't want to be swimming in whole genome sequencing data, there's another smaller bite-sized piece that sequences about 3 million bases in the plant. And that one targets a couple of the key genes. We will sequence THC synthase and CBD synthase and a variety of the genes that are involved in the terpene synthesis pathway, about 30 of them in total. And that gives you some very interesting information around the genes that are governing all of this magic. And then we also sequence another 20,000 markers throughout the plant. And the reason for that is that when people want to do accelerated breeding, this isn't GMO, this is just trying to find the ideal things to cross and to cross them very quickly. What they'll do is try and get a marker every single gene in the genome. And it's probably roughly around 20,000 genes. So if you have a marker for every gene, you can then look for what traits that you're after. Like, are you breeding for large buds and large colas? Are you breeding for drought resistance? So those are things that you want to try and correlate with those DNA markers. And then you can screen for those very quickly the moment plants come out of the ground, and you can accelerate the cycle of selection, where you begin selecting 6 weeks earlier than you might select at the tail end of a harvest. And so this is how most of the agricultural industry runs, is they run off of what's known as marker-assisted selection. It's not genetic modification, but it's just doing what Mendel did with peas at a turbo pace. And we can do this now with the genetic tools we have, and so many people are utilizing that StrainSeek service as a mechanism to track these markers with the desired phenotypes they have.
Now, my understanding is you're not a big fan of believing that there's really any difference between indica plants and sativa. Can you explain that?
I think there once was. A very long time ago, these things were probably geographically isolated in the, Afghanistan and perhaps in equatorial zones.
Kind of like what we would call
the
a
thing
land,
about
like
cannabis-
a landrace strain that is
exactly.
kind of
You might call
right.
them a landrace, right?
Right.
But if you actually study any of the naval records, you'll realize that cannabis was the prior oil. It was what moved the navies around, from both the fiber on the sails and the seeds they would plant wherever they landed so they could grow more fiber for the sails. The oil from the plant turned into a host of different, it's a food source, it's an energy source, it's a fiber source, right? And it grows very quickly. So this powered a lot of the early navies, which means it spread all over the world very, very quickly and got interbred very, very quickly. And what we see now in the genetic picture is something that's very crossbred. You really can't discern indica from sativa, at least in the cultivars that you see here in the United States. Now, you might go down to some very remote regions and start sequencing strains and land races like we've done a few that came out of the Congo, and they're extraordinarily unique. And I wouldn't be surprised when more work gets done in some of these esoteric places that we begin to see a different picture emerge. But at the moment, almost everything that's medicinally floating around in the United States has been crossed over with each other so many times that there hasn't been a single scientific paper that's been able to sequence these things and split apart indica from sativa.
Understand. Well, again, I think I mentioned to you earlier, when I go to dispensaries, and I go to a lot of them, a lot of times I hear the bud tender talking to me about genetics, and it's just cringeworthy because they're talking about, oh, this one's 84% sativa and 16% something else, and I just don't think it really helps the consumer that much, and there's a lot of confusion about that.
No, I think you're right. They need a story. It's kind of like oaky, woody, earthy, and wine.
I got it right.
It's just something that people may be trying to assimilate to one thing or the other. But we haven't had these tools before. In the past, this whole genetic history was recorded as like oral folklore, and that's what we're living through is transforming that into something that's a little bit more precise. But that being said, we don't throw out that oral history. I think that oral history is still valuable.
Sure.
And so we ask people to record it as much as they can, put it into the Canopy entries because there's information content in there that is there's no other way for us to get our hands on it. And so any good genomics project that's looking into the history of cannabis will try and retain some of that information.
Fantastic. Kevin, what role will you play in the events coming up October 22nd at the CannMed Fest?
So I'm going to be giving a presentation on this new genome sequence that we have. That is the most complete genome sequence available for the cannabis plant. And I'm also going to be speaking a bit on how we did this. We did this a little bit uniquely. A lot of people have had a hard time getting cannabis projects funded. The government hasn't been very helpful in funding cannabis research, and so we turned to a cryptocurrency to fund us. And they funded the sequencing of this reference. And we're going to describe a little bit about how that mechanism works to see if other people can try and leverage it as well. And also see if we can touch on where can these distributed ledger technologies or blockchain technologies help in the cannabis industry. We have a banking problem in the cannabis industry. We have a seed-to-sale tracking problem in the cannabis industry. We have people trying to raise money through ICOs potentially. So there's a lot of touch points with cryptocurrencies in the cannabis field. And so we're going to have a session on how those may integrate with the cannabis field. And then there'll probably be some other discussions on microbes and how we can keep cannabis safe by understanding the microbiomes.
Well, I appreciate so much of that because I think one of the things you're really doing is you're not only expanding, the different kinds of applications of cannabis, but you're also helping to produce a safer, cleaner product.
Absolutely, and I think we're finding in a lot of the other agricultural fields that if you don't understand the microbiome, you're going to have a really hard time managing your crops. And this is really critical, we think, for getting terpene expression under control and getting pest management under control, is to utilize biological control as opposed to chemical control that we might be seeing with glyphosate and all these other types of pesticides that are out there.
Now, last question here. So Medicinal Genomics in 2011 had run the genome for the ChemDog plant. That was in 2011. So now it's 2018. So my question to you is, what do you think we're not doing now that we will be able to do in 5 years from now?
In 5 years from now, we will be doing, we will understand the epigenetic code of cannabis. We'll understand all the methylation profiles in the plant, and I think that's going to lead us to understanding the genetic loci of hermaphroditism, to flowering time, to a variety of other features that we've always wanted to understand in the plant. Disease resistance as well. These things are very hard to do with the references we made back in 2011. The references we have now are 400 times more contiguous, if you will. And they're twice as big as well. The entire genome size has nearly doubled since we began sequencing this back then. So once you have a perfect picture of the genome, you can do a tremendous amount of work looking for the genes that are involved in desired, in particular traits that you're looking for.
Well, it's been my pleasure to talk to the folks here at Medicinal Genomics. And again, I want to remind everyone to go to the CannMed. It's CannMed events.com. That's CannMed events.com. And look at this as an opportunity to attend an extraordinary opportunity for those of you who are interested, not just in cannabis, but more specifically in the science behind it. At News Munchies, we're going to continue to support all efforts to reform America's marijuana laws, and I think this is a great opportunity for those who are interested. You can go to the CannMedEvents.com website, and you can include a promotional code associated with News Munchies, our brand, and it's MUNCH, MUNCH 18, and you'll get a discount on the tuition. For those of you who are attending the event on October 22nd, have a great time. Again, this is Ted Corliss with News Munchies. Be well.